Good afternoon, everybody. I hope to entertain you a little bit with some of our research. This is an animal group that a lot of people have interest in it, mainly because they're just so strange. And that's one of the strong points. So I will kick off right away and get us moving. In the top right, you see Woods Hole. We're on Cape Cod, surrounded by water. And at the bottom, you see some of the images of the animals we work with. And my lab does work with a wide range of cephalopods, not just one or two. And I'd like to introduce all my lab and associates who really made all this research possible. I won't go through all these names. They're not all in my lab now. A lot of them have moved on to other things, but there's a wide range of folks, institutions on the left whom I work with. We do a lot of collaborative interdisciplinary work. At the very bottom here, you can quickly see some of the areas of science that we get involved with to study cephalopods. So it's a very integrative approach and very, as I said, interdisciplinary. So let's get going right away and see if we can get a little action here. And I already have, there we go. Now we're diving underwater and there's a back reef area and a fish swimming around and just a rock and nothing happening until now. And you see there was an octopus well camouflaged and it just showed up seemingly out of nowhere. And you can also get other things going here. This is one of the long-armed clade or long-armed octopuses, wonderpuss, that can change immediately and then go back. You see the skin texture is remarkably different. This is a broad-clubbed cuttlefish showing a very strong flashing pattern towards a prey. Here's the flamboyant cuttlefish in great camouflage. And of course, this animal can go to its very flamboyant stage that everyone seems to know about. We call this rapid neural polyphenism. That is, the apparent phenotype of the animal changes dramatically, and it's under neural control, which means it can be very fast indeed. So that's part of what's going on. Well, there's the visually controlled pattern in slow motion, and you see the ring develop around the eye, 5 million chromatophores, another 25 million here, and notch the smooth outline of the body went three-dimensional in rugose. These are called skin papillae, and they are visually controlled as well. And you see that if the animal had kept a smooth outline here, it would stand out against the rugose outline of the algae. So the animals are not only changing color and contrast and pattern and brightness, but they're also getting the skin texture to change in 3D to correspond to the background. And now you see another octopus underwater going against this red kelp, and you see it change its pattern to the texture and the color of the kelp. And this is also visually controlled. We're just going to put it in reverse, and you see the animal looks very different there indeed. And so the changeability is really under control of the animal. I want to emphasize at the beginning that this is not a reflex. This is a decision-making process in a complex brain. And that's really what's going on here. And you see here the skin papillae of the giant Australian cuttlefish. We went from smooth skin to highly papillate skin. And it's like down periscope, and you can see the difference there. And here you have the giant Australian cuttlefish again, and it has a very smooth skin and a very high conspicuous pattern. Watch what happens as I click the shutter of the camera 1 second apart. So we go 1, 2, 3, 4, 5 seconds, and I'm a piece of algae, and then they come back out of it again. And you see that the change in appearance of the animal is truly dramatic. So rapid neural polyphenism, if you look at the top right here, in this squid, it's changing its appearance in 330 milliseconds. A human eye blink is about 200 milliseconds, so this is just a fraction of a second. And another example is down here with the papillae over the eye of the giant Australian cuttlefish, a little bit slower. And down at the bottom, 1,200 milliseconds for this octopus to change its appearance really dramatically. In that case, it's changing thousands of chromatophore organs and millions of iridescent cells and thousands of bumps in the skin all at once. These are all controlled by the brain, so it's pretty dramatic. So how does this work? There's a general cascade of control, starting with the eye. Starting with the eye right here. And there's a big optic lobe, number one, that sends information to the lateral basal lobes, which are the intermediate motor centers and the complicated brain that these animals have. That signal goes down to the lower motor centers of the brain. And this is the important part. The nerve cells have their cell bodies here in the posterior chromatophores. And those nerves travel without synapse all the way out to the skin, to the end organ. These are very long neurons, but they achieve ultra fast speed. So the speed of patterning is much less than a second, as I illustrated. So how does a cuttlefish or an octopus choose the pattern? These animals go out in nature, and I've do a huge amount of field work, and they go to these complicated backgrounds, and they immediately change into what looks like really good camouflage. How did they do that? So we test this in the laboratory by giving the animals different backgrounds. I'm showing those on the left, and we make those with real materials or artificial materials, and we measure all their light characteristics. So we know the visual input and the light input. Then that goes into the cuttlefish's brain, which I'm treating as a box right now. And then the output comes out into some kind of camouflage pattern. And generally, these animals are showing three categories of camouflage: a uniform pattern, or a mottled pattern, or a disruptive pattern. And I'll show you some of those. But if we put a cuttlefish on the bottom into a uniform background, it gives a uniform background. If we change that background to small checkerboards, the animal has changed its pattern to a mottled pattern. And if we take those patterns again and we shift those into large checkerboards, the animal is now shifted from mottled to a highly disruptive pattern, which is dramatically different. So we go from uniform to mottled to disruptive. So the question becomes, what aspects of those checkerboards made the animal switch? And I'll summarize this in a few minutes. But basically, in the third image, the cuttlefish is looking at the white squares, paying attention to them and not the black. And when the white square is about the same area as the white rectangular square that they can physiologically put on their back, they will use that as the cue to turn on a variety of disruptive patterns. So this is the neurobiological shortcut that allows the animals to change so quickly in complex environments. They're looking just for one specific cue for each of the few types of camouflage patterns. That's what we've learned through a lot of hard work. Here's a field image of a very bright cuttlefish, and it just kind of switches down into the sand and turns on different patterns. So it's well camouflaged before and after, pretty dramatically different. In the lab, we can experiment with this. Here's a cuttlefish in a uniform pattern on a uniform sand background. Now we rudely pull the carpet out from under it, and there's a gravelly background. The animal shifts around, quite disturbed by that movement, and then settles into a camouflage pattern. It's got a little bit of disruptiveness there, which it would get rid of if we gave it a moment, but we don't give it a moment. And what you see is our special talented three-handed technician rains in these white rocks from heaven, and the cuttlefish moves over, paying attention to those white rocks and turning on a disruptive pattern. And the idea is that the white rock is much like the white checker, and when it's about the size of the white square in the animal, they use that as the signal to go into disruptive coloration. Now, if the cuttlefish had stayed down here in the mottled pattern, that would have been a great pattern too, and 80% of the visual field would say stay mottled. But there's a priority setting in the camouflage patterns when there's a choice and there are multiple cues in the background. So we've worked all this out with experimentation. So the ultimate goal is to probe the visual system of the cuttlefish to find out what is the algorithm that tells them how to do this. And we do live animal bioassays. I showed you an example there. In this case, the animal is untethered. It's non-invasive. There are no needles or probes in the brain because the neural answer comes out in the skin. It's a visual display of neural activity. Very convenient. And I must mention that the number one defense of these animals is camouflage, so that they are genetically driven to camouflage on every background under normal circumstances. So we can give them crazy backgrounds like checkerboards. They will take that information and do their best to turn on a camouflage pattern. Very convenient experimentally. And we use what's called a psychophysics approach. We not only use natural backgrounds or photographs of natural backgrounds, but we do these crazy things like checkerboards where we can control the exact set of variables, so we can work down to which exact variable the animals are paying attention to. So here's a cuttlefish on small checkerboard. Here's on the big checkerboard. You see the animal. This is 2-speed, 2x speed. And you see the animals turn on a beautiful disruptive pattern because the white checker is about the same area as the white area here. It doesn't matter that it's a checker, by the way. We've done all sorts of shapes and all the rest. It's a light object on a dark background is what they're paying attention to. There also have to be sharp edges, and it has to be high contrast to turn on disruptive coloration. And if it's not, they'll use a mottle pattern. So here is a synopsis of sort of the whole enchilada. Basically, this is how it works. You get visual input here. There's visual processing looking at these factors, the object, the edge, its contrast, its intensity. Then there is a pattern selection process in the upper parts of the brain. And in this case, the parsimonious answer comes out, as I told you, by looking at one cue. And then you get the pattern generation. Excuse me, I went a little too fast there. You get a pattern generation, and a disruptive pattern is made up of 11 building blocks, and they use anywhere from 3 to 11 of those to create a more or less disruptive pattern. I'll point out at the top here, what I'm telling you is based on 25 journal papers, 12 years of experiments on over 800 cuttlefish. So I've summarized a lot here and where it occurs in the brain. That's kind of our status of knowledge at the moment. All right, so how does the skin produce color, and what features that skin really give you this dynamic aspect? And that's really a big question right now. So the skin has 3 layers. There's a layer of pigments at the top. Then there's another layer underneath with iridescence. Those are reflective cells. And then at the bottom, you have these diffusing cells of a bottom base layer of white. What happens is one stray light of photons come in and go through 3 pigments and go out, and you get a certain color. Another bit of light will come in, go through no pigments, will hit in green iridescence. It will come out as green iridescence. And yet another can come down through 1 pigment and go down through an Erytophore and up here, and you get yet another color. So I think you get the idea. With 3 layers and 3 pigments in one of the layers, you can get a lot of color out of that system. Now, it doesn't really work. It's not that simple insofar as, again, we have 3 layers, and working from the bottom up, that white leucophore layer, those are passive cells. They don't move, they can't change. There are no muscles or nerves. When you move up in the middle layer, you have Erytophores. Some are passive, and some are actively controlled by neurons. And when you get to the top layer, those chromatophores open and close, and they're active, very, very active. They're not passive. And so it's the combination of those that give you the answer. Now, I show a little symbol of a smartphone up here, and the reason I do that is to say, have you ever thought about where the color and pattern come out of your smartphone? You've got a battery that you have to recharge every day, takes a lot of power. You've got a computer that's very expensive to create all this light and pattern. And the point I'm making is that our animals are doing all this camouflage and signaling without creating any new light. They are using available light, and they're manipulating light at the wavelength of light with pigments and reflectors. And so a lot of the tools that humans make, or the way other animals produce color, they use a lot of pigments or they use a lot of reflectors. And some animals use both, but not very many. And this is the animal group that changes the most with the most diversity. And we're finding that their secret is using pigments and reflectors in very sophisticated and even dynamic ways. So that's a real biological principle that's emerging out of this. And so here's an example of a kaleidoscope. You see the images just racing around, and I'm just showing close-up images of how different the skin can look. It's a real kaleidoscope of color and shape and texture. And so there's not another animal group that has put in so much evolutionary effort and equipment into the skin, and a lot of genes to control all that skin as well. It's very different from other mollusks. And these are mollusks. They're related to limpets and snails, clams. And it's unbelievable that the coleoid cephalopods are so remarkably different. So back to that skin. It's a neural network in the skin that enables all this. And here you see, here's a chromatophore organ right here with all these muscles. I'll show you those in a moment. You have all these iridescent cells, you can see them over here. And then there are nerves, are the red markings down through here, snaking down. So you got nerves going down through the skin system to implement the control. Here are the chromatophores opening and closing, and you can see they can be very punctate, or the muscles can contract and pull them out into a disc of color. So you have yellow, red, and brown chromatophores. You have 3 pigments in the cephalopod skin. And the chromatophore, when it's closed, looks like this. The chromatophore, by the way, is not a cell. It's a complex organ with 6 cell types. Some of those cell types include the radial muscles that actuate it, and there's a pigment chamber in the middle, and there are sheath cells, and there are nerves, and there are a few other more obscure cell types that make this operate. So if you look here, the pigments are concentrated. That's the case here where you really wouldn't see it on a macro scale. But when the muscles contract, they pull that into a disc of color that you now see. And this can happen, of course, very fast because it's controlled directly from the brain. And it's on the order of 1 to 200 milliseconds that the individual chromatophores can open and close. And the collection of those can go in about 200 milliseconds, as I showed earlier. Now, if you look at this carefully, you see all these nerve bundles traversing down in between the chromatophores. So there's a lot of muscle overlap and a lot of nerve. So this is a very complicated skin. And here again, you see the radial muscles here, and we've highlighted along the iridescent blue here. I don't know if you can see that. Those are nerves latching onto the radial muscles. So there are many synapses along each muscle to make sure that in the moving muscle, the signal gets transferred very efficiently. So let's go to the next layer of skin, the Erytophores, and you see this beautiful blue iridescence down here. And in the very same skin section right here, you see the Erytophores gone from blue to pinky red. That's the opposite ends of the color spectrum. And so how does that happen? Well, these are dynamic Erytophores, and there are nerves traversing that skin. Now we have chromatophores. They're just punctates. You don't see them. And out of nowhere come these iridescent cells, these Erytophores. Watch here how, watch here how the color shifts from red all the way over to green. They're going right across the color spectrum with more input of neural control, and the neurons are dumping this acetylcholine, which is a neurotransmitter. Now, there's a protein inside the Erytophore that reflects the color, and it's called reflectant. And this is a really amazing protein found only in the cephalopod that does a lot with light. And by the way, this system of changeable iridescence under such control is truly unique in the animal kingdom. And now here's a big Erytophore. We stimulate it, and look at the color change throughout here. And if you just look at that again, it goes all the way over to the blue end of the spectrum. So, you know, we've gone from this to this with stimulation. And what it's doing is the acetylcholine is making that reflectant show up, and then the plates squeeze themselves down to different sizes, which reflects the light right across the color spectrum from long wavelengths here to short wavelengths there. It's a very elegant system. If anyone's interested in it, you can contact me. I'll put you onto the literature. Now, there are big neurons right here. This is live tissue in a confocal microscope. And you can see the big nerve bundles here and here traversing down. And you can see all these big Erytophore blotches right here. So the way that works is you got these giant nerve bundles here. Here are the Erytophores up here on the right. And you can see where the arrow goes, all that we've lit up the nerve fibers that branch off the big nerve, and there are nerve fibers traversing all around each Erytophore. And if you look in more detail, you see where the arrows are going here, you see little nerve fibers traversing right throughout that complicated cell. And so this is an unusual system because it's slow. The iridescence comes on more slowly. It takes a few seconds rather than milliseconds. And this is what's called a cholinergic non-synaptic system. It means that you've got acetylcholine is getting dumped out at the nerve terminal, and it has to diffuse into the plates. It's not a direct attachment to a muscle like the chromatophore. So the systems are very different in how they're actuated. Okay, we had a recent surprise, a huge surprise. Look up here, you'll see this iridescence where my arrow is. As we shift the light around, a little hard to see on the screen, but look at this iridescence showing up. Here it is right there. And that iridescence is not supposed to be there. We're looking under the microscope and going, those pigment chromatophores are not supposed to be iridescent. The iridescence is in certain splotches in the skin. Now, here's the skin. And when the chromatophores, look at the yellow chromatophore right here. When the yellow chromatophore opens, you get very specific iridescence that is very specific just to the pigment sac. And so that iridescence is a perfect register with the chromatocytes seen right here and right here. So this convinced us that we found something that all the textbooks said couldn't happen. You don't have iridescence in a pigmented chromatophore. The iridescence is in a different skin layer and different cell types. And suddenly we saw it in the Erytophores. So we're going, well, what is this all about? So we started looking at the chromatophore. This is a confocal microscope image shown by my colleague Steve Semp in my lab. And here's the pigment cell in the middle. And you see the radial muscles going off here. And you see all this green fluorescence all through here, right along the edge of the chromatophore. And that is the reflectant protein showing up. So we're finding the exact same protein that we find in the Erytophores. We're finding it in the chromatophore, spaced all around. And here are just more close-up cells of right here, the green fluorescence. Now we put a red fluorescence on the reflectant and more green here. And you can see that it's all the way around the edge in between where the muscles attach. And then here's the crazy thing that I want to make the point of. These reflectant proteins are not aggregated into a cell or a tissue or anything. It's just tiny molecules spread out all over the place, somehow creating iridescence by a method that, you know, the optical physics world does not know about, and the biologists definitely don't know about. So this is a totally new mechanism of a way to produce iridescence and color. Okay, so these reflectant proteins, as I said, they've not been found in any particular place. Most Erytophores or most reflectants are in some kind of plate or sphere to give a certain color type, not the case. And they're intimately co-located with pigments and reflectors. Meanwhile, we've been working with two reflectant proteins that we've known about. The Squid and the Octopus genomes have just been finished. And when you look at the genome, we find 18 reflectants in the Squid, thanks to Carrie Albert and Dan Roxar and associates who did the genome work. So this means essentially that we've really been working in the dark, pardon the pun, because there's a lot more iridescent possibilities in the skin, and different reflectants are going to light up in different places to help us understand where this reflection structural coloration is coming from. So there's a lot more to learn, and this is all very new information. We're really excited about this and hoping to get some funding to study that. Now, cephalopod vision, which controls camouflage and signaling, just tell you a brief thing about, you know, the octopus eye or the cuttlefish eye, squid eye. They have good visual acuity. They can really see things sharp. They have an eye that, you know, at least grossly looks a little bit like a vertebrate eye. They have excellent night vision. We've done studies on this. They can see very well and light that we're completely blind at, that is true starlight, 0.003 lux. Those photographers who use lux will know that's dark. They have very good contrast sensitivity, but crazy enough, they can see polarized light. We can't see polarized light. It's because their photoreceptors are arranged orthogonally. And of all crazy things, they're colorblind by anyone's experiments or any knowledge we have. So, you know, among other questions is, how does a colorblind camouflage coordinate its color for good color coordinated camouflage? Well, I'm going to reach that subject in just a moment. Here's an octopus moving along on an Okinawan reef, and it's very conspicuous, and it just goes in there and stops. And the pattern match and the color match and the contrast and brightness match, I have full framed it right in front of the camera, and you really cannot discriminate that this is an octopus. It's just another rock or piece of coral on the reef. This is why when you and I go diving, we can't find the boogers. It really spends a lot of time trying to find the octopus in the first place. And when we find one, you know, we film it carefully at a distance, but we don't take our eye off it. So what is it doing, and how does it do that color match without color vision? So there's before and after. So we've been thinking about this a lot. Now, here's the same species of octopus on a different reef. This is not a color match. They don't color match every moment they're on the reef, just most of the time, which we're now measuring from field data. Cuttlefish can do the same thing. Here's the giant Australian cuttlefish, which really is a big animal. They can get up to 5 or 10 kilos. And this one's only about 2 kilos. But watch the same animal as it descends to the bottom and does that. Do you see the cuttlefish right here? I hope not, because the cuttlefish is actually right in the middle and made it easy for you, but it's really hard to see that cuttlefish again. So the pattern change and everything has changed, and it looks color coordinated. Now, is it? Well, we're looking at RGB images tuned to human vision. So, I'm going to show you later on that we use other instruments to try and process this so we can see what that looks like in the color spectrum of predators, not humans. I'll get to that in a minute, but the color match is looking pretty darn good. And I've done thousands of dives and have a lot of data. And to me, a human color match looks pretty good. Not that that's what counts, because we're not shaping their evolution. Okay, so Colorblind Camouflage, this beautiful paper by Justin Marshall, John Messenger in Nature back in '96, showing in an experiment how they might match the background. So very speculative, and that spurred on 2 detailed papers by my lab, published in 2006 and 2010. We got evidence for some light sensing in the skin. So if the eye doesn't have the material for color change, then maybe the skin can do some of that. And I don't have time to talk about this paper right here, but we essentially took things like those checkerboards I showed you with the cuttlefish, and we made the checkers different colors and made them exactly the same brightness so that only a color-capable animal could tell yellow from blue, and they couldn't do it on different contrast. And the poor cuttlefish just thought that was a uniform background, so they could not perceive it when you only gave them a color cue. So meanwhile, and now I'll summarize 5 years of work by 2 large labs right here. The bottom line is that we don't know how they do colorblind camouflage, but we have established light sensing molecules in the skin. And these are the opsin molecules that are in the cuttlefish or octopus retina, just like we have opsin molecules. Those are the ones that transduce light energy into a neural signal. And we found them in cuttlefish and squid in great numbers, and Todd Oakley and his group in octopus found them also, but we couldn't really make a correlation to whether or not they can use that skin information of light to adjust their pattern. And so it's still an open question and a really difficult one to study. So we don't know the answer to the colorblind camouflage yet. It's one of the things we still have to learn. Now, in terms of color and camouflage and how do they get the pattern right, we need to figure out ways to study color and quantify it under natural light conditions. And we've been doing that with our RGB cameras. But technology really is an impediment because we need cameras that do something other than RGB and capture more colors. Just capturing 3 colors, red, green, and blue in every pixel, which is all of our instruments do, tuned only to us. There are 300 colors technically in the visible range between 400 and 700 nanometers. So we've gone to the trouble to design and build a hyperspectral imager, which has 16 colors in every pixel. And so we published that initially a few years ago, showing that if you put a cuttlefish on a background here in all these images, you can look at that in different ways from different eyes of different predators. And so this is the test you take at there's a numeral 5 right here, numeral 5 right there. I hope most of you can see it. If you can't, you're colorblind to some extent. So if you're monochromatic, but this is what you do at the driver's test. We did the same thing to analyze what this cuttlefish might look like to different predators, one of whom might have, this is what we look at in our RGB world, that's for humans. We could look at a predator with 2 color receptors, a dichromat, one with 3 like us, but different ones, or a monochromat. So that was a first step, but it wasn't a very good one because we were in the lab and no natural light. So we went to the trouble to create this new camera that has 16 colors in every pixel right across the range, visible range, including a little bit of UV. And we took this to Raja Ampat in Indonesia, where you have incredible color variety in coral reefs. It's thousands of square kilometers of pristine coral reefs. And so this is where we went and took our special camera to try and see what's going on. So here's an example of the three of us down there. Here's the hyperspectral camera and its big thing. And we're just now beginning to analyze some of these data. I don't have time to show it to you, but the question is, when you get an image, what does it look like to a different predator? So this is guesswork at the moment, but a colorblind animal like an eel would just see the black and white thing and wouldn't see that octopus in color. A dichromatic animal like a barracuda might see a little more color. A snapper, which is a trichromat, a little bit like us, might see it a little better in this color field. And a tetrachromat, like some diving birds and a few other animals, very few have 4 color receptors, but a cormorant is one of them, would see more color. So we're trying to figure out how to visualize and analyze the data, and that's ongoing literally as we speak. So we don't have the answer, but we're starting to get there with some better technological toys. Now, you put this all together with correlated behavior. Here's a perambulating octopus, goes across an open area and goes into this fabulous moving rock trick where they bundle themselves up when they're nearby rounded things like rounded corals, and they look like another piece of coral, and they actually walk very slowly. Now, when there aren't rounded rocks around, here's the octopus right here in Puerto Rico. When there are large spiky things in the area, like out here, and they're not small and rounded, the animal now takes its arms and puts them up, puts its skin papillae up, so that we call this the moving algae trick. So the behavior and sort of the cognition of the animal to look at the surrounding area, even distant objects, so that it becomes a masquerade of those other objects, is one of the parts of camouflage that these animals do so elegantly. So I'm going to finish up the talk and talk a little bit about the flamboyant cuttlefish. And we just published this paper a matter of weeks ago. And this is camouflage tactics and colorful reproductive behavior. I want to tell you that one of the driving forces to do this study was that the internet and public aquaria and many stories are all about the flamboyant cuttlefish. Oh, it's so colorful and flamboyant, and it's really amazing. And that's all you see on television. Well, we're diving on this animal in the natural habitat, and the story turns out to be really different. This is an example of what that cuttlefish is doing when it is flamboyant. It will do this. These are a series of drawings by my co-author, undergrad at Northeastern University, Gwen McManus. And they take their arms and they do this bowing thing in the courtship display to females. And here it is right here. And I'm going to show you the video. And here's the small cuttlefish on the bottom right. And here's the female not paying any attention. Now watch this little character. He's putting on the bright coloration, moving around, intercepting the female, and doing this dynamic patterning that's very high contrast and very colorful. And this is really dangerous because these animals are susceptible to predation, and they're making themselves more visible to predators. And indeed, during our experiments and watching these animals underwater, we did see one of these small males. And by the way, this is the normal sexual dimorphism. The males are very small, females are much larger. We saw a small male backing up, doing his bowing routine to the female, and he got snapped up by a scorpion fish. So there is a trade-off to this signaling, as there are in all sexual systems. Okay, watch. This is a big female here, and here's a guarding male. This is a larger male. There's actually a small male right here, and I want to point out that these guarding males doing mate guarding, and they're actually fighting the small male. The fighting males will also put on this dynamic flamboyant patterning. So watch this little guy darting in. He's really aggressive. And the guarding male is staying between the intruder and the female. So it's a very active process. Notice this little guy just darting around 180 degrees, trying to get in different ways, and the guarding male is very effectively blocking them. And they're really exposing themselves with that bright pattern again. So this is where the flamboyant pattern is being used, and it's not being used very often. The really full-time camo is the name of the survival game in this animal group when we studied them. And here you see some examples in the top right. Believe it or not, this right here is the flamboyant cuttlefish. And this is some glop underwater, and here's some more glop underwater. And the cuttlefish just came, settled here, and took on the pattern and texture. This looks nothing like an animal. The same cuttlefish can go down to sea and put on a disruptive pattern, or it can go over here and put on a mottled pattern, or like the bottom one, it's a mottled pattern. Now with the video lights on, we've brought sunlight down here, and it looks like the color match is not very good. But under natural light at this depth in the bottom right, when the color is not there or it's muted, you see that the camo match is really very good. Okay, so just to wrap up, the cephalopod eyes have a couple of simple rules to guide dynamic camouflage. We've shown you experimentally how that can work. The skin is absolutely built for speed and diversity. And these animals have a wide range of pattern repertoires, mainly for signaling and fighting. That's for sexual selection, but also for camouflage. The secret to the skin diversity are pigments and reflectors being used in sophisticated ways. And I will point out that there is an untapped gold mine among cephalopods and some fishes, which is unstudied, of how pigments and reflectors are used to create more optical diversity, and a lot of color mechanisms there to be discovered. Color perception is very difficult to figure out. We don't know how the animals might do that. There are a couple interesting leads, but no worthy information yet. We hope this gets studied in the future. The flamboyant cuttlefish is not often flamboyant. That's the bottom line of our paper and the very extensive field work. We did 2 long-term studies in Indonesia, diving studies with 15 volunteers in each one. There was a huge effort. And so our data, our field data, are pretty good or trustworthy. So I end up by saying, so many questions, so few answers. Isn't this typical in biology? As soon as you learn one thing, you figure out how much you don't know. So one last thing I'll just say at the end here is my colleagues and I have published 2 books recently that have a lot of this and other information in them. One is Cambridge University Press book on cephalopod behavior. This is a very rigorous scientific document, every chapter peer-reviewed, but John and I wrote the whole book. We've reviewed well over 1,000 journal publications on cephalopods, and that's the 2nd edition. And then we did a more coffee table book for the public over here on the right. But I point out we made it very scientific. It's filled with scientific information, but many more huge pictures than the other book, which is also highly illustrated. And I give a lot of credit to Mike Vecchione at the Smithsonian and Louise Alcock in Ireland, and my co-authors who really did more than their share of the book. So it's really great to acknowledge their work. So with that, I'll sign off. I always point out this saying at the top, which was put in the MBL library here in 1890 by Alexander Agassiz, study nature, not books. That's one of my excuses to go diving. So thank you very much, and I will be happy to take questions.
One question we had is, the cuttlefish appears to have its eyes on the top of its body. So how does it see the surface below itself to kind of know what to work with?
It does not use the information directly underneath it. It uses the information adjacent to its body, because that's the information that a predator would view the cephalopod in. So just remember that the cephalopods all have their eyes where our ears are. They do not have binocular vision. So they have monocular vision. Every once in a while, they can scrunch their eyes forward, but they only do that during feeding. So with one eye on either side, they're picking up the visual information adjacent to them. We've actually measured the field of view that they're using to get that information. And generally, it's 1 or 2 body lengths out from them. They use that nearby information for that visual microhabitat to decide on their pattern. And that makes all the sense in the world, because they have to camouflage, within that area first. So that's the best I can tell you about that question.
I know from reading that I've done that there are a lot of practical applications that have come out of research on cephalopods for things like soft robots and things like that. For your particular area of research, do you know of any real-world practical applications that are being developed?
Yes, I would tell you that on all of our grants and projects, we have material scientists and engineers as collaborators on the grant. Um, there are many desires and applications for changing color and pattern of our clothing, our cars, you name it. And those capabilities, for the most part, do not exist. And so our collaborators are looking for ways to take information from how chromatophores and erytophores work and how to translate that information to making human devices. Um, as you might imagine, and if you may have noticed in my second slide, I am funded a fair amount by Department of Defense because camouflage is important to our soldiers. And hunters are very interested in this, and so are SWAT team members and others. So not being seen has some human functional significance. But beyond that, there's also the soft robotics community. We are currently studying work right now on the octopus arm as an example of the most flexible appendage in biology. And we're looking at the color change capability of the arm, the sensing capabilities of the suckers. We're proving the degree of flexibility and figuring out how that's controlled from the brain. Each individual arm has a little miniature satellite brain that controls some of the sensing and motor activities of an arm, but it all has to be coordinated by the central brain, and a lot of study. Soft robotics, there aren't soft appendages well developed by industry, but you can think of a lot of examples of crushed buildings trying to deliver drugs or radios to people who are in crushed buildings. If you want to look at the human system, you can think of the whole GI tract and tracheotomies and the other end as well. Wouldn't you love to have a more soft, flexible appendage doing that? And so you can think of a lot of ideas about what to do with soft appendages. So there's another example. And then finally, just in the world of cosmetics, I talk about this a lot, making changeable cosmetics that use pigments and reflectors in more sophisticated ways, the way that animals do. And, you know, I gave a TEDx talk at Estee Lauder, the cosmetics company, and I told them that, you know, this is possible. We have enough information about how animal pigments and reflectors work and how they can be controlled that you could start to translate this information. So, you know, I don't want to hold my breath, but I'd say in the next decade, you're going to start seeing things like that, where ingenious people take these ideas and sometimes the actual pigments and do things with them from animals. And, you know, so there are a few examples. I could go on, but there's a lot going on there.
Okay, so we had actually a couple questions from college students. We've got a student from the UK who's actually in her final year for undergrad. She wants to know, for someone that wanted to work in this field, what would they go into after they completed their undergrad work?
Well, I think in a graduate program, you would want to concentrate. There's sort of two ways to go about this. If you want to know about the behavior involved in camouflage, you want to learn ethology, which is the biological study of behavior. But if you want to study the skin, you really want to learn a lot about chemistry and pigmentation. If you want to learn about the dynamic aspects of color change, you need to learn a certain amount of neuroscience so you can understand how nerves can operate things. And in general, cell biology, because most of these things are happening at the cellular level. So there are many levels. On the other side of that is to look at biooptics and learn the physics of light, because that has a lot to do with how these pigments and reflectors work. So there are many ways to do this. And in graduate school, I always say, you know, this is biology, so you want to learn as much biology as possible, but as much chemistry and physics, and in this case, optics, that you can learn, you can really do and address much more important and more detailed and difficult questions. So I said a lot of things there for the person in the UK, but there are many ways you can go there. So I wish you the best of luck there.
Okay, so I want to go ahead and follow up with a question from a grad student currently working on octopus arm movement study. So the work they're doing is apparently recording signals as they travel down the arm. And they're asking how electrically active is the skin on these animals?
Well, the skin is very electrically active. There are, in a squid or an octopus, there are tens of thousands of chromatophore organs every one of those chromatophores is innervated from the brain, and there's a lot of neural tissue. So there's an enormous amount of neural activity for the skin alone. And those skin papillae that I talked about, those bumps that go up and down in the skin, they are all controlled by neurons also that control the muscles that make those bumps go up and down. And so just for the skin alone of an octopus arm, there are a lot of neurons controlling the skin patterning. And the skin patterning on the arms is just as refined as every other part of the body. Now, in addition to that, you can dig into the big nerve bundle going down the middle of the octopus arm, and it's absolutely enormous. There are many nerves in it. But if you go down to each individual sucker on the octopus arm, and there are 100 to 200 suckers on every octopus arm, multiply that times 8 for the animal, each sucker has 10,000 neurons connecting to it. And many of those are for taste and touch. They're sensing organs, not just grasping organs, those suckers. So you've got an octopus arm that is just chock full of neurons. So there is a lot going on, and it's very difficult to tease those out. Almost every one of those, many of those have cell bodies in that ganglion at the base of each arm. I glibly called those little satellite brains. They aren't really brains. But some of the skin ones are almost certainly going all the way back up into the brain. The brain, the central brain, is probably controlling most of the chromatophore neurons on the arms, but others are probably out there in the periphery at that little satellite brain. I'm talking about a very complicated animal, the octopus. I mean, all of the three groups are complicated. The octopus might be the weirdest and the most complicated. It's a hard study animal. We're working actively on that area. So I can tell you that it's not easy. It takes luck to make a breakthrough and a lot of hard work.
So I'm curious about, how do you find these things when you're on these dives if they camouflage so well? I mean, is it just you've gotten used to being able to pick them out, or is it just you get lucky? Or
well, there's some luck involved. I'll tell you how not to do it, and that is to just swim around on the reef and kind of glance around. That's not going to work. I started out like that, and I was swimming over octopuses and getting nothing. You often start at their den. These animals live in the den most of the day and night. And so if you can find their den, where they're living in the den, some octopuses are real litter bugs. Octopus vulgaris, very common on Caribbean reefs, takes shells and crab carapaces. They bring it back to their den. They eat it. They spit it out. And so we look for depressions in the ground where the litter bug has left some shells out there. That's one of the best ways to find where an octopus lives. And when you find it, you can shine a little light in there and see if it's there, put a little marker on it, then come back the next day early. And this is what we do with divers, or we put a small camera or remotely operated vehicle there, and we wait until the octopus comes out on its own the next day to do one or two of its forages. And when it comes out, then we have divers posted nearby who stay at a distance and wait until the animal habituates to the diver's presence, and then the octopus will go out and start foraging normally. That's when we start getting data. So we burn out a lot of volunteer divers because they're down there watching a hole in the ground, and nothing happens for an hour, and then the next team comes down. It can be really tedious. But when you do that and the animal's moving around, that's when you get your data. And so it's not easy. Sometimes you get lucky and the animal will be out and then it'll try doing an attack and it'll flash a pattern and you can find it and then you follow it that way. Or you use local guides all the time at dive operations. They know where to find the animals and how to find them. We always work with them. And then the thing is, I take 15 volunteers out on these trips at Citizen Science, and they're all searching for animals in holes. And then we find one, we set up a team, and we get our scientific data. So it's a key question, and it takes a lot of time up front just to begin to get data.
All right. Well, we love citizen science here at the Science Museum of Virginia. We have run a couple of projects. We're actually running one, running an air quality research project right now. So here's a question. Does it tire the animals when they're using their camouflage capability? So when you're researching them and they're changing colors, does it tire the animal at all? Does it fatigue them when you're doing this?
No, I've never seen that in all my years of doing this. Plus, we culture the animals in the lab. They're using their chromatophores day and night all the time. So the related question is, how energetically expensive are all those chromatophores? And the skin probably is energetically expensive, but there might be a solution. No one studied it. We don't have physiological data, but other mollusks like clams have a wonderful evolutionary development called catch muscles. And catch muscles are muscles that can almost go into this tetanus-like contracted state, and they can lock there physically so that they're not expending ATP to stay contracted in the muscles. And catch muscles are a very molluscan thing. They're in a few other phyla, but they're not very widespread. And so people are just starting to pay attention to this. You know, we wonder if the skin papillae, which are these great big bumps, they don't tire of those either. We wonder if there are catch muscles there. We have some indirect evidence that the right neurotransmitters are there that are used on catch muscles, but that's not very good proof. It's just the first step. So a great question. I've never seen them tire out, but I think there might be a trick there to keep them from burning out. I mean, all these cephalopods are all living a year or less. There are just a few rare exceptions. And so it's live fast and die young. Everything these animals do seems to be energetically costly. If you think about a squid is negatively buoyant, and it literally has to pump just to stay up 24 hours a day. An octopus is sitting on the bottom, can be much more restful and conserve that energy. And a cuttlefish has got a new solution. The cuttlefish, which is negatively buoyant, has a cuttle bone, and they pump gas in and out of it to stay neutrally buoyant at whatever depth they are. So that's a really cool invention by the cuttlefish. So the squid hasn't figured this one out yet, but squids don't live very long either. So the energetics of a squid, just a little bit mind-blowing to most biomechanics folks and physiology folks, because they are really, really working hard just to stay up in one place in the water column. So we know a little bit about that, but never enough.
All right, we have time for one more question. Does the animal's internal state, like sleep or feeding, change their ability to change their color or their pattern?
Well, we don't know too much about sleep. We did publish a paper 2 years ago on sleep in cuttlefish, and we showed what could be considered REM sleep, rapid eye movement sleep. During that sleep process, the eyes were shifting all around. Most of the chromatophores were closed, and then weird patterning would show up and then turn off. Nothing normal patterning. And, humans tend to say, well, that's what my dreams are like. There are all kinds of incoherent things flashing in my brain, and it looks like the cuttlefish is doing that. So there does seem to be all these animals, the cuttlefish and the octopus is definitely sleep. We don't know about squids, maybe not, but we don't have any data showing that sleep will help them learn how to use their camouflage or skin better. The other thing is that when a cuttlefish or an octopus hatches out, right out of the egg, like the flamboyant cuttlefish, the full repertoire of camouflage is in place and they're doing it right away. So they are innately developed enough so that the camouflage is there right away. That hatching stage is the most vulnerable to more predators. And, I glibly describe an octopus or a squid as a yummy hunk of protein swimming in the ocean that everything's trying to eat. And there's some truth to that phrase. They're right in the middle of the food chain. And so this idea of having your camouflage working right away is an important one, and we find that in most cephalopods. Now, these animals have short and long-term memory. We know that. They have spatial memory that's very well developed. They have huge, complicated brains. We know where the memory is formed in the brain. So why couldn't you use short and long-term memory, you being the cephalopod, to improve your camouflage in different habitats? That's possible. There's only one study out there. It's a little controversial. It didn't have all the controls we would have liked, showing that in the first 3 or 4 weeks of a cuttlefish hatching out, if you give them an enriched background versus a plain boring background, that the animals seem to camouflage a little faster on different backgrounds in a test after 3 weeks when they had a lot of visual diversity in the background. It doesn't exactly answer the question, but it's the only study out there that we have to talk about that. But fundamentally, the capability to camouflage is built in right near the beginning, and the animals really do a great job. During my postdoc many decades ago, I actually studied that in cuttlefish from hatching all the way up to adult size, and I showed that how cuttlefish work from the babies on up. And it turned out that the babies in the young stages actually have better camouflage than the larger animals and the adults, in cuttlefish at least. And that's because they're more susceptible and they don't have that large size to help depend on getting away from predators.
All right. Thank you so much, Dr. Hanlon. Great presentation. Thank you very much for being here. So it looks like we're out of time. Thank you all for joining us. So please join us next week, November 25th at noon. I will be your host next week. I am a historian by training, and science history is kind of my specialty. So I will be presenting I Wonder What's in It, How Science Made Food Safe. And I'm going to be talking about the history of the Pure Food and Drug Act, and how chemists were actually able to stop food adulteration and make food more safe. So I hope you'll join us for that. You can register for that talk at smv.org. Each talk is free to attend and is open to the first 300 registrants. Thank you again for joining us today. Until next week, stay safe and stay curious.